pesticide use, and chemical fertilizers, two well-known, commonly accepted agricultural practices. For a long time, they have boosted and stabilized our crop production, but there is also a flip side, a loss of biodiversity. And not only above, but maybe even more so below ground.
We found that in croplands, mycorrhizal biodiversity is lower on average. You have fewer species.
While in wild soils, these microorganisms are still there and are collaborating with plants to protect them against the stresses that in agriculture we control with pesticides and fertilizers.
An ancient corporation that used to have the same function as fertilization and pesticide use today.
So when we started doing the domestication, the first thing we did was do land clearing. So we remove all these plants we didn't want, and we just leave the ones we want. Remove all this plant diversity, reduce the diversity also in the soil, in the microbes, without even us knowing about it. And with this reduced diversity, plants start needing more help from us. So we start to applying more nutrients, we start to applying more pesticides, more insecticides to recover all these things we lost by the ecology. We put it back, bad input as humans.
And that's why in agricultural soils, many of these protective microorganisms have been lost.
But there's another way.
In MiCRop, we want to understand the exact differences between these wild and agricultural soils. We want to look for lost microbes, microbes that are still there in these wild soils, but not anymore in agricultural soils, and that have maybe important functions that could also be useful for our crops. And on the other hand, we are also looking into our crops, our modern genotypes. Have they perhaps lost the ability to collaborate with these beneficial microorganisms? We have maybe been breeding crops that are making less efficient use of these beneficial microorganisms. We believe in order for agriculture to become more sustainable, we need to go back to this ancient collaboration between plants and these microorganisms.
Through unique research,
Mapping plant microbe symbiosis, mostly for mycorrhizal fungi, across spatial scales.
The cool thing, but also the disadvantage of these fungi is that they form super thin hyphae, mostly in the soil. So if you just look at soil directly, you won't see how they grow, you won't see how they interact. We are trying to get the fungus to grow in non-sterile but clean, transparent environments.
We look at the plant defense system from a different angle, not only at blocking pathogens, but also how it enables plants to accommodate beneficial microbes in the roots.
How the artificial selection of characteristics in plants and the effect of agriculture in the soil have changed how crops today interact with the microbes.
And what is very special about MiCRop is that we're studying this in 100 different plant species, crop varieties, and wild relatives of these crop varieties. And in all these different plant species, we're looking at how the microbiome is protecting these plants against stress.
So the idea is to recover these ancient microbe alliances the crops have in the centers of origin and translate them to the agricultural setups we have today.
we try to shift perspectives.
From being reactive to a pest, shifting the focus into leveraging interactions with the pool of beneficial microbes that are already in the soil and strengthen the plant's own defense machinery against pests.
These underground maps are very surprising. They're showing that the majority of mycorrhizal infrastructure, these threads underground, are in ecosystems that you don't think of as being relatively important. So grasslands are really important ecosystems, for example. But our above ground bias means that we think of them as less important compared to something like a tropical rainforest. The goal of these maps is to flip that on its head. Imagine if you had the first map of the Earth's forests. That's what we're trying to create for below ground life.
Traditional approach will, for example, look in the plants for resistant genes, so genes that will make the plant resistant to the pests. And from the other side, biocontrollers will look for inoculants that can attack the pests. Now the idea is not to look for these resistant genes, but to look for the genes that calls for the microbes, and the microbes that are necessary or missing that can respond to this. So then you have a proper communication between microbes and plants, because plants now produce the messages they need, and you have the microbes that are ready to understand and hear what the plant is saying.
If all tools have been biased and built to understand symbiosis from a plant perspective, you're missing half the story. Right now is the time where we're building microbial tools to also understand the below-ground perspective.
And join forces to integrate research data that can benefit agriculture and nature alike.
Have a collection of microbial genomes that people can use, for example, to combine later for their creating smaller synthetic communities to test specific questions. Other datasets that were generated, different stresses were explored on different parts of microbe, and that gives a big overview of how microbial distribution, let's say, changes across different plant species, but also across different stresses.
With that knowledge, we will be able to make use of this collaboration with the microbiome to have an agriculture that is making less use of chemical fertilizers and pesticides, and that means less damage to the environment.
And not only in Western agriculture, but in the entire world.
The reason why we teamed up both with Western companies and with international institutes is because we believe that the knowledge that we generate in MiCRop can not only benefit agriculture in the West, but can also help make agriculture in the developing world more sustainable and more resilient against stresses.
This really brings together that MiCRop is, at the end, about the farmer and their field. It is thinking about how can we improve crop resilience for actual people in the world.
The final mission of MiCRop is to secure a stable food production both in the West and in the developing world, but without the negative impacts that all these synthetic inputs have on the environment.